All right, my name's Mike Hickerson, and I'm from City College with Anna and Kyle, so I'm really used to the hug fest. It'll be a surprising part. I'm going to keep it down 3 minutes, but so a lot of what drives my interest in biogeography and ecology and evolution is really realizing this pipe dream of what I call the pipe dream of comparative phylogeography, kind of inspired by John Haynes. The idea here is bridging phylogeography with ecological theories of phylogeographic assembly. And what do I mean by phylogeography? My way of looking at it is basically using genetics, population genetics specifically to reconstruct species histories. And the pipe dream really is, if you can do that, well then let's look at multiple aggregate species histories, aggregate population genetic data sets, and try to understand better the assembly of communities. And up until now, the last 20 or so, 30 years, this in practice has really been just quantifying the distribution of species responses to past historical changes. So thinking about things like different colonization trajectories into the northern temperate regions, or looking at the variability in isolation or expansion times, bottlenecks across species. But the general idea has really been almost like really theory free, really quantifying the distribution of histories. So what we've been more recently been trying to do is actually linking a lot of these models with ecological theories. So specifically, classic models like MacArthur and Wilson's theory of island biogeography, Hubble's neutral model, Tillman's competitive assembly model, and so on and so forth. So the idea here is really building these models of community history with ecological theory. And I would like to just kind of, here's a current kind of project realization here. Andy brought this up in his talk here. This is the MESS model, Massive Eco-Evolutionary Synthesis Simulations. Now the idea here to bring out is that this is a process-oriented model linking processes underlying community assembly with patterns of several dimensions of biodiversity. So we have the macro evolutionary phylogeny patterns that this can predict, different processes, spectrums of genetic diversity, and also spectrums of species abundance. So the idea here is we can actually take data and then go back and test whether particular assemblies are conform to sort of neutral models or non-neutral models. Is competition important? Is environmental filtering important? And then maybe we could actually take this to a third dimension of the actual remote sensing. Can we actually predict different patterns of remote sensing? Okay.